to just thank the National Park for inviting me to come speak, and thank all of you for taking time in your evening to come and see the results. So as the introduction, my name is Jen Caselle, and I'm a research biologist up at UC Santa Barbara. And typically, I would start a lecture like this, or I'd start a presentation like many of my colleagues with a whole list of doom and gloom, the problems. What are the problems? The oceans, the health is bad, the marine fisheries are declining, pollution is increasing. Ocean acidification is going to melt everything in the ocean or boil it, or they're going to boil, or animals are going to do both, melt and boil. And, you know, I was putting this together and I decided I'm not going to do it. I'm not going to do it tonight. We're all aware of the problems in the ocean, and I'm not trivializing them, but I'm just not going to do it. Maybe sometimes us scientists do that in some way to justify the work that we're about to do or to show you. So what I want to do tonight instead is I want to celebrate. I just want to make this a celebration of things that the state of California has done, that our local communities here in Santa Barbara, Ventura area have done regarding marine protected areas. A lot of people have put a lot of hard work into designing and setting up MPAs. People, scientists like myself in agencies, in National Park Service, in the sanctuary, folks in environmental groups, the fishermen, fishing communities. And so I think I just feel like tonight we're going to just celebrate some of the efforts there. So with that in mind then, I'm just going to skip the ocean is going to hell in a hand bucket introduction and start right in on what is a marine protected area. Oh, I guess my own little clicker may not be working. Let me see if I can turn it on. I may be stuck behind the podium here tonight. There we go. Okay, so what is a marine protected area? There's been a lot of activity in the state of California, and I'm sure many of you have heard about marine protected areas, but if you haven't, they're basically areas, geographic areas that are set aside and limit some form of activity. So it's always about, it's a spatial form of management. It's always about an area. There's a lot of different terminology going around, MPAs, marine reserves, and so forth, especially globally in the global community, but California at least has sort of landed on several definitions. In general, at the highest level, when we talk about a marine protected area, usually some forms of activity, often fishing, are allowed in marine protected areas. But if you hear the term marine reserve, that almost always means it's a no-take, no extraction whatsoever. In California, we've got 3 designations: state marine conservation areas, which do allow some recreational and commercial activities, and that includes some forms of fishing. State marine park, where certain recreational fishing only is allowed, and then a state marine reserve where all fishing and all resource extraction is prohibited. So these are sort of the designations that you'll see in the media and when you read about MPAs in California. But they're just areas that we set aside to limit activities. There's a lot, also, as many definitions as there are for marine protected areas, there's many different goals and reasons for setting these places up. And this big, huge run-on sentence characterizes pretty well all of the different goals that one might want and some of the reasons that we set up MPAs. So the important bits in between the many commas are in the colors. But basically, the goals for marine reserves or marine protected areas: conserving biodiversity, improving management of fisheries, preserving and restoring habitat. That pretty well summarizes what MPAs are about. Why do we want to do those things? Well, society wishes, in some cases, to preserve areas simply for the enjoyment of nature. Let's not forget that that is a perfectly valid reason. It's not all about maintaining fisheries or producing things. Sometimes we just need places that are wild. To maintain functional ecosystems, marine protected areas set aside an entire area. It's the habitats and it's all of the species within it, so the whole ecosystem. Establishing replenishment zones for overexploited overexploited species, so fisheries enhancement is a potential reason as well. And also to provide insurance against the uncertainty inherent in managing living resources. Managing living resources is really not an easy thing to do. The environment is very variable. It's hard to predict, especially with the backdrop of climate change. So these are places that provide us essentially insurance. Okay, so lots of different goals. MPAs are definitely a tool that's used worldwide. This map is just an example of a study that some colleagues of mine did where they, and I'm going to show you the results of this study in a minute, where they took data and results from MPAs and all these different red dots, and they summarized it into one easy, digestible figure. But they're a worldwide conservation action. So I will put on my little pessimistic negative. I'll go back to my doom and gloom introduction just a little bit. So this is a slide that I would have been in the doom and gloom introduction if I was giving the doom and gloom introduction. They're really rare in the sea, less than 1% of the ocean in marine reserves. Here's a global map. 5 years ago, this was the state. You couldn't even see anything on the map, can you? Because they're too small, okay? Literally 5 years ago. But that's not really what's going on anymore. There have been dramatic changes, and this is what the map looks like now. However, not to be totally positive, it's still estimated that only 2.8% of the ocean is protected in some form of spatial management. That's not a lot, and it definitely is, and nowhere near the global goals that the IUCN has set of conserving and preserving 20% of our oceans in some form of marine protected area. So it's not great, but at least you can see them on the map now. So it's better. Okay. Part of the reason you can see them on the map is because there has been this trend towards, and it's a pretty recent trend, towards implementing very, very large MPAs. I call it the arms race because presidents of countries are trying to outdo one another by protecting giant areas. Our own president recently expanded protection here. Perhaps some of you read about it, the Pacific Remote Islands National Marine Monument. And it held the record as the largest protected area for about, I don't know, I think maybe 6 months before, breaking news, the UK passed up the US and designated complete protection for the Pitcairn Islands. So it's not even on this map because it only happened last month. And it is now at 834,000 square kilometers, the largest of the no-take, complete no-take protected areas. So it's 4 islands around Pitcairn. Remember, some of you might recognize Pitcairn, Mutiny on the Bounty. Bounty mutineers went there. Really crazy, fascinating. So the UK protected the entire Pitcairn Islands exclusive economic zone, and it's quite a large area. So this is a recent trend. I like it. It's an arms race that I can live with. There's also been an increased call for establishing networks of MPAs. As single small MPAs were put in place all around the world, and some positive results were starting to be seen, people have realized that putting together regional networks might be a good idea. So what do we, and that's what we've done here in California. It's what we did in the Channel Islands. So what constitutes a network of MPAs? It's just really just an integrated system of multiple protected areas. These can be more or less designed scientifically. In California, they were very well designed scientifically, or at least a lot of science went into designing them. Networks meet a lot of the same goals that I read to you in that giant run-on sentence. So regional biodiversity and ecosystem function. Also, buffering against catastrophes even within the network. If one MPA is wiped out for some reason or something happens, you have a network that is supposed to be self-seeding. From an ecological perspective, a good network would be designed so that the MPAs are connected via larval dispersal or even via adult movement of organisms in them. So it becomes sort of self-replenishing. And I will note, because it's not talked about very much, but there's other kinds of networks beyond an ecological network. Social networks are important. So creating a network of MPAs can create social dynamics around, or social cohesion around that network as well, organizing the people. So today, I just want to show you, as I said, in sort of, it's a celebration for me in a way, the results of a fair amount of hard work over the last decade in the Channel Islands Marine Protected Area Network, which was put into place in 2003. So just a sort of brief outline. I'm going to show you a lot of data. And although I think we're going to do Q&A after the presentation is over, if If anyone has a specific question about what I'm saying, you can't follow the graph or I didn't explain it very well, could you interrupt me and stop so that, because if you're confused and I'm not explaining it well, probably everyone in the room is. Okay? We're going to talk about changes inside and outside the MPAs in the Channel Islands. We'll look at various species groupings and single species. We'll talk about the potential for MPAs to affect directly areas outside the MPAs. Okay? And then we'll talk about some of the changes that may occur in MPAs that have to do with the entire community shifting and how our kelp forests will look inside the MPAs. So that's just a really rough outline. So when I showed you that global map with all those red dots on it and all those little numbers, and I said that some colleagues of mine did what we call a meta-analysis, and they took results from all over the world, summarized it into one easy-to-digest plot. This is what they found. It's probably not that surprising when you remove the effects of fishing from a given area. You will see a buildup in density and a buildup in biomass. Biomass simply means the total weight of animals in an area. Most of the data I'm going to show you tonight are also fish biomass. So it relates to how many fishes are in an area and how big they are, because it's the total weight. Okay? So biomass, much, much greater in MPAs, density greater, size of individuals a bit greater with lots of variation, and diversity, which means more species in MPAs as well. So although this does show that on average the effects of MPAs can be quite large, I do want to point out every one of these dots is a data point from a different study. Look at the amount of variation. And in many cases, there are examples where density was lower inside MPAs. So it's not that simple of a story. But on average, the effects are quite large. So that's sort of talking about species and populations in MPAs. I'm also going to show you some results about spillover. So the idea, I'm taking you kind of through the outline now, and then we'll get into the results of the Channel Islands. Reserves can also affect populations outside their boundaries through something we call spillover. Spillover is exactly as it sounds, right? Spilling over outside the boundaries. And it can happen in a couple different ways. It can happen through egg production or larval production. Now, most marine animals produce pelagic larvae or eggs. That means that they go off swimming into the blue. So you can get larval spillover by big individuals growing up and having a lot of abundance inside a reserve and contributing larvae and eggs that then drift outside of the MPA and they seed areas outside. I'm not going to talk about that tonight. That is a very difficult thing to measure, larval spillover. We're working on it, but it's, as you can imagine, a very difficult thing to measure because larvae are the size of this big in a big ocean. Adult spillover is a little bit more tractable of a problem, and the idea behind this is that as biomass or density builds up inside of an MPA, juveniles or other individuals might find this area inside the MPA too much competition going on, or perhaps all of the habitat is taken, and they will immigrate to outside areas where there is more available space and less competition, perhaps less predation. So that's the idea, is animals moving outside of the MPA. And then finally, as we move down in our outline, is this third idea, the third concept, that reserves might affect the entire community structure within an MPA. And the way that might happen is through shifts in food webs because of what we call trophic cascades. That means buildup of predators eating the prey and all these dynamics that will shift what a community looks like in the area. This is a really nice example that comes actually from the Channel Islands. It's from the old Anacapa Reserve, the one that's been in place for 30 plus years now. Some colleagues of mine at UCSB. And the idea, it illustrates nicely this concept of a trophic cascade. In the reserve, there were lots of lobsters. Lobsters eat urchins. In the reserve, there were few urchins, but outside the reserve, there's lots of urchins. And urchins eat kelp. And inside the reserve, there's lots of kelp. And outside the reserve, there is not, probably because the urchins eat it all. So this is a really simple a very simple food web. And I'll show you an example of some really new hot off the presses work that we've done that illustrates some pretty cool stuff going on possibly in Anacapa. Okay, so that's a little background to the concepts that we're gonna go through. So here's the setting. I'm only talking about the Channel Islands Reserves network today. Most of you are probably aware that this is now set within the context of a statewide set of marine reserves that have gone in through the Marine Life Protection Act, but I'm not going to talk about that today. Those reserves are very new and we are monitoring them, but we don't have the time series that we have from the Channel Islands. I also don't have Santa Barbara Island included in this map because I don't have a lot of monitoring data from Santa Barbara, and it won't be although it's part of the national park and it's part of the Channel Islands National Marine Sanctuary. I won't be showing you results from Santa Barbara today, just from these islands. So to situate you, Anacapa, Santa Cruz, Santa Rosa, San Miguel, with the different designations of reserves delineated on this. So before we jump headlong into data, I just want to give you a little bit of background. I'll go back over here to the podium so I can go through this really quickly of the timeline and how did we get to where we got, where we are now basically in the Channel Islands. So it started back in 1998, and the Channel Islands Marine Restoration Committee, which is actually just a group of concerned citizens, including some spear fishermen, folks like yourselves, they made a request to the Fish and Game Commission that the commission establish a network of marine protected areas around northern Channel Islands. So the Northern Channel Islands MPA network was a citizen-driven event from the very beginning. It was not legislated like the Marine Life Protection Act is. And that's, in my mind, important. So then some things took place. 1999, the Channel Islands Sanctuary, the SAC from the sanctuary, and CDFG established the Marine Reserves Working Group, affectionately known as the MRWIG. And for any of you who sat on the MRWIG, I'm sorry if this is going to bring up really old nightmares and so forth, because the MRWIG met every other month between 1999 and 2001 to discuss planning and mapping and where and if MPAs should go into place. So when I said in my introduction that I wanted to celebrate a lot of people who did a lot of hard work, those people who sat on the MRWIG were champs, okay? Unfortunately, the MRWIG never did really come to a consensus. Not all processes come to consensus, but they did come to a point where they could make some recommendations to the sanctuary for implementation of MPAs. It was also 99 that this state legislation, the MLPA, passed that told the state to designate a system of MPAs, although that one took a while to get going, so we're not going to talk anymore about the MLPA. This is also when my group began monitoring, when PISCO was first funded and began our program of monitoring. It should be noted that the National Park Service, of course, as I'm sure all of you know, have been monitoring for a really, really long time, much longer than the PISCO group. And we're working together now to put our data sets together so we can have even longer time series. In 2003 then, that is when the Channel Islands network was established. There were 11 SMRs, reserves, no take, and 2 SMCAs that allow partial take. And the sanctuary and my group PISCO and the Park Service and the Fish and Game, which is now called Fish and Wildlife, hosted a workshop up at UCSB in 2003 to talk about and design a plan for monitoring and to create priorities. And so this was a really very sort of consensus-driven process the whole way. The plan was created in 2004. We jump ahead to 2007. The MPAs initially went in only in the state waters, and in 2007 they were extended into the federal waters. And the final network then includes 318 square miles now, currently, just in the Northern Channel Islands. 2008, happy birthday, 5 years old. And we celebrated with a 5-year evaluation that was semi-formal. This went to the Department of Fish and Wildlife. Those same partners that I talked about earlier, the sanctuary, the park, PISCO, the department, held an evaluation. We held a public symposium. There's an informational booklet, and we made presentations to the commission. And the early results were pretty preliminary. 5 years is not a long time for change, but there were glimmers of positive change at that time. I said I wasn't going to talk about MLPA, but it should be noted that in 2012 then, in the Southern California area, this MLPA network of MPAs was implemented. And that was tricky because the Channel Islands were pre-existing. So the Channel Islands MPAs are now included in the MLPA set of MPAs in Southern California, but there was no move to redesign them in the Channel Islands. Thank goodness, right? That's a problem for monitoring when your MPAs move around in space. So, problem for a lot of things, actually. Okay, so this gets us to almost now. Network was 10 years old in 2013, and at least from my group, our monitoring data were beginning to be long enough to allow time series, and here we are. This is actually the map of the entire Southern California area and all of the different MPAs that are in place now through the MLPA. And again, we'll just be talking about here. So, okay, so I better move along. So how do we know if MPAs are working? And I should have put working in quotes because working depends on what you define at your questions to be, what do you want out of them? Well, the answer is we monitor. And there's a few points I want to make about monitoring. Monitoring is super important. These MPAs in California were supposed to be, they are adaptively managed. That means that we need to evaluate what they're doing and how they're working, and if they're not meeting their goals, then there should be discussions about adaptively managing them, which may be changing aspects of design. So monitoring is super important for that. The sooner we can determine the benefits and the costs, the more rapidly we can refine the design. There's also another issue that isn't really so much a problem in California, but it is definitely a problem globally. That is that if you put these MPAs in and they actually are ineffective, but you're not evaluating them, you're giving a false sense of security. And in many places in the world, this could actually jeopardize the resource, especially if other regulations are relaxed on the outside. So that means you come in, people promise big things from an MPA, other regulations then get relaxed and the MPA doesn't work and you don't even know it. That is not happening in California, but I put it up because it is happening globally. And from a nerdy scientist perspective, I mean, these things offer us the potential to see how humans are affecting marine ecosystems, how systems are going to behave in the absence of major human pressure. This is something that, you know, is important. We want to know. Oh yeah, and then there's the legal requirement as well. So we need to monitor. So today I'm going to focus the talk only on kelp forests. Kelp forests are very important ecosystems in worldwide, really. They're important here in the Channel Islands, especially. They have very high productivity, and they also provide for very important fisheries. So really all we're going to talk about today is kelp forests. This is a little chart showing the productivity. Productivity is measured in how much carbon is produced. Look where kelps are. I mean, they rival coral reefs and tropical rainforests. Those are things that people tend to think as like super highly productive environments, right? Kelp forests are right up there. I'm not sure that that's really known by most people. So we do, my group does scuba surveys. It's very similar to what the Park Service does in their monitoring programs as well. And I mean, I'm not going to belabor this. We just basically go out there. We spend a ton of time underwater. We count everything. We estimate sizes of things. We spend a lot of time training to do it. And it's really fun sometimes on days like this. Will it work? I rarely do videos because they never work. One of the things that is really important to monitoring is that we monitor. We don't just go and monitor inside the MPAs. We also monitor outside the MPAs or reference areas so that we have something to compare to. So, you know, it all looks great, right? And we love what we do, and especially on days like this. So it's not always as easy as it looks. This is a new diver in training. We didn't count this transect. Don't worry, we don't count fishes this way. Poor guy, headed off in the wrong direction. Getting his butt kicked. I think this is at Miguel on a particularly, there's his trainer kind of floating above him. And if you had audio, you could actually hear us laughing on the camera. So yeah, takes a fair amount of training, and you can see him shaking his head like, oh really, are you really filming me? And that's what we do. Here's where we do it, all through the Channel Islands. We have sites, as I mentioned, inside and outside. I told you already, I'm not going to talk about Santa Barbara Island today. But it is critical that you monitor inside and outside because so much of what we do, you have to compare. This is a very changeable environment. Things change naturally. So in order to know if things are changing due to the effects of an MPA, you have to know how things are changing naturally in the environment. We do that by measuring both inside and outside of MPAs. The other really cool, yet really difficult thing to manage in the Channel Islands is that the Channel Islands spans a really, really large gradient of environmental conditions. Who's made dives in the Channel Islands? Who's made dives from the east to the west, or at least in the east? And so you know how different it is. When you're in this map right here shows a 20-year average of sea surface temperature. So basically, it's warm in the east. And it is really cold in the west. And what that causes is really different community structures. The fish that you see up here in the west look like the fish you see in Monterey, and the fish you see down here in the east have tropical affinities. So what we have to do in all of our analyses is we have to essentially factor out the environmental variation so that we can test the effects of reserves, right? And so I'm not going to talk too much about how we do that, but we do it in all of the analyses. Here's an example. So our state fish, the Garibaldi, it's 92 times more abundant in the east than it is in the west. It doesn't really even occur in the west. Kelp bass, one of my favorite organisms, 84 times. Here's a species that's only in the west. It's 235 times more abundant in the west than the east. So we have to take this into account when we're measuring the effects of MPAs. Okay, so I know, let's get to the results, right? So given the biogeographic differences across the islands, and you'll just have to trust me that we factor that out through various statistics, what do we want to know? We want to know, I mean, do you want to know? What do you, yeah, we want to know. Does the density and the biomass differ inside and outside of reserves? And now what we really want to know is how are those patterns changing in time now that we have a time series that's long enough? So one other way that I'm going to show you the data is to simplify things, is to group them into groups of species that are targeted by fisheries and groups of species that are not targeted. The reason we do this is we have a hypothesis that the species that are targeted by fishing, those are the ones that are likely to respond to an MPA, right? They're the ones who aren't getting fished in an MPA anymore. Species that are not targeted by fishing, we don't have hypotheses really for how they're going to change inside and outside an MPAs because they're not harvested. There may be changes in the community structure, and we'll get to that at the end. So what have we found? Well, this is the figure that I showed at the 5-year review of the Channel Islands, and here's how it works. It's biomass of fishes. These are the ones, targeted species, targeted by fishing. These two bars over here are the non-targeted species on the right. And red is inside the reserve and blue is outside the reserve. Even after 5 years, we saw significant differences with more fish biomass inside the reserves for the targeted species than outside. I think actually at 5-year mark, we were pretty surprised only because we didn't think things were going to change that quickly. And here's the non-targeted species. As we hypothesized, we didn't see a difference between inside and outside. So now that we have 5 more years of data, here's what the patterns look like. So these are the averages from 2003 all the way to 2012. And basically, it's just the patterns that we saw are continuing and are even stronger. So we have an even larger, more significant difference in the targeted species with more biomass inside than outside. So it wasn't an anomaly at 5 years, and our hypotheses for, at least for the targeted species, are confirmed, and things are changing, and they're continuing to change. Okay? That's overall the average. I already told you about all that environmental variation, though, so is this happening the same way on all of the islands? The answer is no, it's not. Now what you're looking at is targeted species on the left plot, and the forget the non-targeted for now. Let's just look at the targeted. Here on the left plot, red is inside reserve and blue is out, and here are the islands. Anacapa on the right here, Santa Cruz, big differences. Santa Rosa, big differences. Much more fish biomass inside and outside, but no effect at San Miguel at all. So we're seeing stronger responses in the warm areas and less in the cold areas. Now I'm going to leave it to you guys. We'll get back to why that may be. I'm going to let your brains go and think, well, why would that be? What about the environment? What about fishing pressure? What about things like that? So think about that, and we'll get back to it. Okay, Here's the plot we couldn't make at 5 years. Here's the plot that we can finally make now with 10 years of data. This is the time series. Starting in 2003, going to 2012, the targeted species are here on the top plot. And again, let's focus on the targeted species. In the red are inside marine reserves. And look, there's a lot of variation. I mean, there's some up and downs, but it is a positive trend. Biomass is increasing in reserves. But wait, check out targeted. These are species that are fished outside of reserves. There's also a slight positive trend. They're increasing as well. They're not increasing as fast as they are inside MPAs, but they're certainly not declining. This is a really important result because one of the things that could have happened and was a big concern and a valid concern was that basically things would go to hell in a handbucket outside of reserves because all of the fishermen have to redistribute and they have to go to areas now that are open for fishing. And that could cause a lot of pressure on the fish species outside reserves. And we're not seeing indications of that in this data set. So I find that very, very positive. Again, non-targeted species, we don't have strong hypotheses. They're no different inside and outside reserves. Okay, but everybody in these talks always says, but Jen, I like sea anemones, or I like red urchins, and they always want to know about individual species. So we're gonna have a little audience participation here. Here's a whole bunch of species. These guys over here tend to be the fished ones, lobster, rockfish. Over here are things that are not fished or maybe lightly fished like kellet's well. Who would like to see a particular time series? Garibaldi, okay. So our state fish, the Garibaldi. Here's how all the plots are gonna look. I know, I'm gonna walk you through it. They're by island, and sorry for the blurriness. I don't know what went on with the program. Here's Anacapa, Santa Cruz, Santa Rosa, San Miguel, clockwise. All the plots will look like this. Red is always inside reserve, blue is always outside reserve. So a couple things with Garibaldi. First, remember that geographic variation I was telling you about? Garibaldi don't really exist in the West. That's why there's nothing on these plots. You can ignore the San Miguel and Santa Rosa. Let's look at the trends in Anacapa, for example, or Santa Cruz. When these shaded confidence intervals overlap with one another, that means there's no difference in those trends. So basically, as we would suspect from a fish that is not fished, there's no difference inside and outside of reserves for Garibaldi at Anacapa, where they exist at Anacapa or Santa Cruz. There's also no strong positive or negative relationship. Okay, so they're all gonna look like that. Somebody tell me another favorite species. Lobsters. Hello, Mr. Lobster. Same thing. I'm gonna give a big caveat here. Visual surveys, the way we do them, it's not necessarily the best technique in the world for surveying lobsters. We do a pretty good job, but we don't go out at night, we don't trap. So there's my caveat. The data are still valid, they're still real. So we don't see a lot of lobsters at San Miguel and Santa Rosa. So again, let's focus on Anacapa and Santa Cruz. Okay, here's Anacapa. Some interesting things going on here. Inside of the reserves, there's a lot of variation. Look at the dots, they're all over the place. But in general, there are definitely a higher abundance of lobsters inside reserves than outside. Santa Cruz is interesting. Santa Cruz appears that lobsters are increasing dramatically inside of reserves, whereas they're not declining outside of reserves, they're just holding steady. Could be taken as a positive indication. I'll tell you, I do a lot of this diving myself, and I will tell you that these reserves at Santa Cruz, there's lobsters all over the place, and they're out and about, literally going like, you can't catch me. And the other ones are hiding. So, okay, we could do a couple more of these, even though I have spoken way too long already. How about we do one more, and then in Q&A we can do, oh, my favorite, sheephead. I'm so happy somebody picked that. I do a lot of research on sheephead. Here's the sheephead trends. They do occur all the way across the islands. They're kind of the poster child a little bit for MPAs, but we have seen some very strong increases in the biomass of sheephead. Their numbers are increasing, but what's really increasing dramatically is their sizes. So strong increases at all of the islands, except again, San Miguel. And no real pattern of decline outside, maybe a little bit at Santa Rosa, but they're holding steady outside, but they're increasing inside. Okay, so I would love to do more individual species, and especially happy that my little animation thing is working, because I wasn't so sure about that, but we'll do more in the Q&A, so if you have some individual species you want to see, we do need to move on. Okay, I thought I had hidden these. We don't need to do these slides. There's another way that we present single species data, and it's in the form of ratios. So what the response ratio is simply the biomass inside versus over the biomass outside. So the way you read these plots is if the dot is over to the bigger than 1, that means there's more inside reserves. And if the dot is to the left or less than 1, that means that there is more outside reserves for all of the individual species. You don't have to read these individual species. What I've done is I've color-coded them by whether they're fished. Targeted in this case is targeted pretty heavily. Incidentally fished are things like surf perches. They might get caught, but they're not super targeted. And then unfished. Take-home message here is that most of the heavily targeted fish are far more abundant inside than outside. If you look at the kelp bass, it's almost 3.5 times greater inside reserves in its biomass than outside. And this is the same plot for invertebrates. Similar sort of pattern. Spiny lobsters, warty sea cucumbers, red urchins are the more heavily targeted species. Turban snails and kellet whelks are not heavily targeted in the islands. They are sort of on the mainland with more abundance inside. And those species that are not fished tend to be right around the 1-to-1 line. That means they're about equal inside and outside. So I'm going to move on. I'm going to try to take about 5 more minutes and just give you a couple little stories about, one about spillover and one about community changes. We defined spillover already, right? The thing about spillover is that spillover can lead to some very interesting patterns around the edges of reserves, on the boundaries. The reason is that if you look at this sort of pretend little cartoon, if fish abundance, in the red, if they're in the MPA and they're in high densities, and as I said, due to maybe high predation or maybe there's no food, a lot of competition, they might be moving to the outside areas. You can sometimes see a gradient of abundance right on a boundary like this. So you can see a pattern like that. You know what else you can sometimes see? You can see a pattern like this. If, for example, the fisheries are doing something that we call targeting the line, which is a really smart thing to do if you believe in spillover, your highest catches will be right on the boundary. You can actually end up with patterns like this. It's a gradient, but with a decline around right on the boundary and an increase after. So what we do is that, and this is work I did with the Park Service, we set up a few reserves around the Channel Islands where we put a lot of sites inside and outside. This is scorpion. So we have all the sites inside in the red and lots of sites at varying distances outside from the boundary as well. And then we look at those fine scale patterns. And we look for what I just showed you, like in those cartoons. We did this at actually 4 reserves, but I'll just show you some results from the scorpion reserve. Here's the density of sheephead. And it's similar to that pattern, that cartoon I showed you, right? Maybe possibly some fishing the line going on. It's, you know, it's hard to interpret, but definitely higher abundances in the core of the reserve and lower abundances to the outside. But then, you know, abundances get kind of high again as you move away from the reserve. That was sheephead kelp bass. Not as clear a pattern, but again, kind of a drop-off with definitely a higher densities in the core of the reserve. So if we sum up all the different, that's just 2 individual species, but let's look at our targeted species again. The way this works is, here is the center of the reserve. Can you see the red shading? That's the reserve. So the edge of is the boundary there and the boundary there. For the targeted species, all that whole grouping of targeted species, these are the biomass values inside the reserve, and they are definitely lower to the outside. Although once you do this kind of aggregation, it's hard to see any kind of fishing the line patterns or detail like I showed you in the cartoon. Okay? So, and no pattern at all for the non-targeted species. Again, that shouldn't come as any surprise given what I showed you in the first section. Okay, that's all I'm going to say about spillover. Finally, what about this community structure business, this trophic stuff? Now, community structure. The changes are a little bit more difficult to measure, and also we think that the changes in community structure are going to take more time than those simple changes in abundance for various reasons. One is that to get changes in the community structure, we need the whole life cycle to happen. We need recruitment of new individuals into the area. But also, to get changes in community structure, we have to be able to measure these species interactions, competition and predator-prey interactions, like I set up in the introduction. So I just want to tell you, actually, I want to skip this because I've taken too much of your time, because I want to tell you this story at Anacapa that we just have been figuring out through observations and just putting into data. So, and then I'll end with this. So Anacapa is interesting because there is an old reserve here, 30 plus years in place at the Landing Cove area. In 2003, with the new designations, there was a new State Marine Reserve, so this is a no-take, in the Middle Island, and an SMCA was put into place in the Western Island. And this SMCA allows lobster fishing, both commercial and recreational, okay? So we have old reserve, old no-take, and new partial take. So let's look at those main players, the urchins, the kelp, and the predators of urchins, and see what's happening. I'll walk you through this. Let's look at Landing Cove first. Landing Cove, in the green is kelp, and in the purple is purple urchins. Purple urchins eat kelp, remember? So at Landing Cove, and we've known this from the Park Service data as well, historically, virtually no purple urchins, really consistent kelp beds, okay, in this old reserve. But let's look at the new reserve. What's happening here? Well, we've noticed over the years that purple urchins have been declining and kelp has been increasing in this new no-take. However, over here in this new SMCA, we have not seen that pattern. We're still seeing a big abundance of purple urchins and no giant kelp at all. So what's going on? Well, let's look at the predators of the urchins and see what they look like. There's two in particular, the lobster and the California sheephead. In the lower plots, the California sheephead is in red and the lobster is in blue. In Landing Cove, the old marine protected area, there's a lot of variation, but there's still really high abundance of both lobster and sheephead. Presumably, they're keeping the urchins down. What's going on in the new no-take? Well, look at this. Both predators, with a lot of variation, but both predators are increasing over time. So they may be resulting in this decline in purple urchins. Over here in the SMCA, the California sheephead is increasing. It's not allowed to be fished there, but the spiny lobster is not. It's allowed to be fished there. So this is setting up a really interesting dynamic. And what we may be able to figure out is what's going on over here. It may be that both predators are required to keep those urchin densities down. Perhaps just the sheephead alone is not enough. Perhaps the lobster is the more important predator. We don't know. This is pretty new stuff. And we're watching carefully. There's one little thing I want to say that some of you may ask me questions about later. There's also an invasive seaweed that's coming in, right? Called sargassum. And we do not see sargassum in the old reserve, where there is a well-established kelp bed and lots of understory algae. Here, as these urchins are declining in the Middle Island, we do see an increase in kelp. We're also seeing an increase in sargassum. We've removed, the herbivory is being removed by the urchins, and now it may be that those algae are duking it out. And so we don't know what's going to happen there. We also see sargassum here. Or we don't see sargassum here because we still have a lot of purple urchins. So crazy mystery, intrigue, crazy story of predators, kelp, urchins, and so forth. So hopefully I'll be able to update you on this sometime in the future, and we'll know what's going on. But it is a great example of how entire communities can change. So I'm going to summarize briefly. I'm not going to read all of this to you. One thing is that, can you hand me those things right there? The results that I told you, we just made a new little pamphlet. Unfortunately, I don't have very many because I just didn't print very many, but they're available as a PDF on our website. So all of the results are summarized in here. So if you really need a paper copy, I have a couple here. But the real take-home message here is this, that for me at least, we do see the effects that we observed at 5 years. They're continuing. They're even more pronounced. But the big one for me is that the biomass of the fish targeted by fishermen is increasing both inside and outside of reserves. That's important to me, that things have not declined greatly outside of the reserves. Again, we didn't have a lot of hypotheses for the non-targeted species. Invertebrate species that are subject to high fishing pressure, especially sea cucumber, urchin, lobster, are also more abundant in reserves. But in some of those cases, they're actually showing declines outside of reserves. So a little bit different than the fishes. Let's go in a little more detail with this. I said I'd get back to you. I said I asked you to set you a task to think about some of these things. So this business that things are not going to hell in a handbucket outside the reserves, it suggests to me then that this fear of the shifting fishing effort really making big declines, that it's not happening, and it hasn't overtaxed a lot of the fish species that in these open areas near the boundaries, and that again is really important. We do need to do a lot more work, though, to understand whether the increase is related to changes in the fishing patterns around the MPAs. It could be due to spillover. It could just be changes in ocean conditions, although then you would, if it were just that, you'd expect to see the same patterns inside and outside of reserves. It's probably a combination of all of these factors. And finally, the spatial variation. We didn't see strong effects at San Miguel. MPAs are in colder waters. There's a couple reasons potentially for this, but it also requires more work. It may have to do with less fishing pressure up in Miguel compared to Anacapa and Santa Cruz. It may have to do with the fact that the species up in that colder water tend to be slower growers and long-lived. So, okay, so I have talked way more than I had intended to, and I've taken a lot of your time. Hopefully you will want to stay for questions. I do just want to end on some acknowledgments just of our funders and all of the different partners. This work can't be done without the sanctuary, the Park Service, the Department of Fish and Wildlife, some of our funders, and also about a gazillion divers, literally lots and lots and lots of divers. So I'll end there and take any questions, or we can go back to single species as well. Yeah. I think they want you to speak into the microphone so that you're going to be recorded, just so you know.
You mentioned before that the sheepheads were increasing in size. Do you find this across all the species, that they're getting larger?
Yeah, and we do. So one of the first things that we see is just when you remove fishing, you see the existing fish that are there are able to grow larger. That's one of the early effects of an MPA. In order to get more numbers of fishes as well, then it requires that whole life cycle again. So you've got to have input, right? You've got to have recruitment. Recruitment is really variable. So there are other things that can make the increase in numbers lag behind the increase in sizes. That's one of the reasons we use biomass as a metric. It accounts for both, essentially. It accounts for more numbers and bigger fish. Okay, well, maybe here for, oh, Bonnie's gonna choose the.
I'm curious about the red sea urchin.
Yeah.
As the individual.
Do you want to see, you want to go back? Okay, good.
Yeah.
Go back to the individual plots, especially now that they actually worked, my little hyperlinks. Sea urchin is interesting case as well. So let's go here. Okay, red sea urchin, it's harvested quite a lot. There's some interesting things going. It's not super abundant in the east anymore. Looks like there's no difference really. There's if anything, there's more outside of reserves at Anacapa, slight declines inside. No real trend at Santa Cruz. And Santa Rosa, we don't see a lot of red urchins at Santa Rosa. San Miguel is a really interesting case. There's some weird stuff going on there. In San Miguel, the reason for this huge increase in the reserves is really due to one location only. It's a location near Hare Rock that turned into a red urchin barren especially in about 2008 is when that big jump up was. And it's been persistent there. There's no kelp. It's unusual for red urchins to form barrens like purples do. We've had a lot of discussions with the fishermen, and nobody really knows what's going on. They're not particularly good to harvest because they're in a barren, so their roe aren't that good. It's one of those things that it's a weird case, and I wouldn't say this has to do with necessarily just protection. There's a question here, Bonnie.
Oh, thank you.
I was just curious about the data for the targeted species outside the protected areas.
Yeah.
Does that come from the fishermen themselves? How do they report that?
No, these are just surveys. Everything I showed you comes from our fisheries independent visual surveys. So these are all just scuba divers doing transects and so forth. So, but that's a really good point, okay? I'll make a point on your point. And that's that we really do when I say it doesn't look like the fish are declining outside the reserves, it's just the scuba surveys. I'm really not able to say, well, and that means the fishermen are doing great, or the economics of the party boats are great, or that, you know, the lobster fishermen are making a ton of money. I really would not extend to that. We really need more study of the socioeconomics of what's happening to the players in the system, the human players in the system. So how, you know, how's the fisheries, how's the economics going? Are people out of business? Are they not? So this just, you know, and that's why I kind of phrase this as things are not tanking it. They're not, in our data, they're not tanking it. And if they were tanking it, we would be able to say they're tanking it. But that's just the animals. I kind of stick with the non-human animals. They're easier.
I was just curious if you noticed any changes in maturation rates in juveniles to adults with the associated increase in mass, and if so, did you see maybe a lag time associated with population growth in any species?
So that's a good question. No, we haven't in the context of the MPA monitoring because we haven't actually done those studies. Some other work that I've done on California sheephead definitely have indicated very strong spatial variation in growth rates and time to maturation. And then it's really interesting because those guys are sex changers as well. So there's strong geographic variation in the time to sex change and the sex ratios. But we don't know about differences inside versus outside of the protected areas. We would expect that to happen after either A, the biomass has built up so much that there's competition going on, or something that would expect, you know, that we would expect to cause a change in growth, and or B, that the so biomass building up, or that the community structure has changed completely and the same prey aren't available, again, giving us some idea for why growth rates would change. So those are studies that should be done, and I think the reserves are probably mature enough now to start doing them. Yeah, unfortunately, those techniques are usually require us to kill the animals, and we try not to do that inside the marine protected areas.
Have you seen the effects of sea star wasting disease and the effect on the that has on the environment?
Totally, yes. So these plots all kind of ended in 2012, right? I have data through 2014. Sea star wasting has happened. Have people seen this in the news? It's supposedly, I mean, my colleagues say it's the biggest mass die-off in marine ecosystems ever, like ever, here on the west coast of the US. The sea stars we see it. So as of last year's surveys, we basically do not see any sea stars anymore except for one species, which happens, it's called Pateria, the bat star. It tends to be more of a detritivore. So one species in particular that has completely disappeared from the western channel is the sunflower star. And the big sunflower star is also one of these urchin predators. You know, I told you stories about lobster and sheephead. Well, I should have included sunflower star, but I really didn't want to go there because there aren't any left. So we suspect that there could be follow-on changes to the communities with the loss of that urchin predator. We don't know its effects relative to sheephead and urchin and lobsters. We think sheephead and lobster probably eat more urchins than the sunflower star is capable of, but there should be changes in the community structure. You know where the changes are going to be really dramatic is in the rocky intertidal, big time, where those sea stars are keystone predators. And you're going to see all sorts of interesting things, probably shifts in elevation of mussel beds and so forth. You need the microphone.
I've also read recently that the sea urchin population is suffering as well. Have you heard anything about that?
I'm super glad you asked me that because I'm kind of a little bit, I'm trying to get to the bottom of that. I'm not happy about that article. So one article was written about, it was a really interesting article. It was on the Nat Geo blog site or something like that. And it indicated, and then it was picked up by a lot of media outlets. And it indicated that we should be watching because the urchins are dying, and it gave the impression that it may be the same pathogen, and now they're just going to follow in the footsteps of the arm steps, of the sea stars. And so I've been doing a lot of research on that just in the last week or so to get to the bottom of who's reporting all these urchins dying. And basically, that was probably just a neat story that writer wrote. We do see, I don't know how else to put it. The urchins are not dying, everybody. The urchins are not massively dying right now. But let me give you some facts. Yeah, mass die-offs of urchins happen. They've happened in Southern California a lot. There's a world expert on it right there in the third row, Dan, who's probably seen a lot of it. And it happens. And it usually is associated with warm water, which is what's happening right now, okay? But we aren't seeing mass urchin die-offs right now. There's always a low level of disease in urchins. We always see urchins with, even in cold water, we see urchins with little missing spicules. We see them with these black, necrotic little things on them. And there's always a low level of that disease. So I've polled David Kushner, who's the lead on the kelp forest monitoring program from Park Service, who watches for these things and is one of the best naturalists that I know, and also my colleagues down in all throughout Southern California. No one is seeing mass urchin wasting right now. I'm not saying it can't happen, and even if it did, the guys who have been studying the pathogen in the sea stars say it's super unlikely that it would be that pathogen anyway. So take that article with a
We have
grain
time
of
for
something.
2 more questions.
In the past couple of years, the price of lobster has gone up a lot, and I've noticed about twice as many lobster traps in my favorite dive areas. Has the population of lobster changed any that you can tell in the last year or two?
Well, our data from these reserves definitely indicate lobsters are increasing inside the reserves, but that's not, the guys aren't trapping inside the reserves. I don't really know, except probably similarly to you, I have lots of buddies who are lobster fishermen, and things have been pretty good. It's a well-managed fishery. There's a lot of communication within the fishermen themselves. There are limits on how many traps. I don't remember what it is offhand. There are limits to how many trap sets you can make. So that's all I can really say is things have been pretty good for the lobster fishery the last few years. Yeah, they recruit on warm water. So this year probably brought an influx of recruitment that's going to help out for years to come. Yeah.
And any final question for tonight?
Did you have another question?
Finally, the article that I read about the urchin had something to do with Fukushima, and that might have been a factor there. And I was wondering,
did it really? You're serious? Did the article imply okay.
And so they're saying
see that
they're
one.
seeing this further up north. But you say you monitor these things. Is water quality and these kinds of things also monitored and the effect upon these animals as well?
Water quality is monitored, not by us, by others. And the effect of water quality on organisms is that linkage is usually sort of indirectly made with basic water quality standards. Certainly the beach watches link water quality to us, to our human health. The Fukushima thing, people are measuring radiation levels in both organisms and looking for it in the water. And there's no evidence at all that those levels of radiation are there is evidence that through the isotopes that, for example, there was a really neat study from some Stanford guys that showed that there were the specific isotopes that could be linked to Fukushima in tuna, and tuna crossed the entire Atlantic. So, and Pacific, both actually, they're big migrators. But that's different than talking about levels of radiation that are harmful to human health. So they did not find any, and no one has found any radiation that's at the levels that could be harmful to humans. I was involved in one study on the West Coast right here locally where a researcher at Cal State Long Beach was organizing all of us to collect and submit giant kelp, because giant kelp will take up those isotopes as well. And we were meant to do 4 or 5 collections over a period of a year, and he stopped after the second collection because the techniques to do it are expensive, and not a single blade of kelp had any evidence of any radiation. And that was from Washington down to Southern California. So there wasn't really any point in continuing the collections. So, Yeah.
This concludes our lecture for tonight. We just want to say a big thank you to Dr. Caselle for joining us, and thank you to all of you.